This video is sort of like a preface to my upcoming video on the paper VSEARCH, a versatile open source tool for metagenomics. This paper is best understood if we tackle a couple of terminologies mentioned. So let's get started. Metagenomics is the study of genetic material recovered directly from environmental samples. It is synonymous with environmental genomics, ecogenomics, and community genomics. Metagenomics is both a set of research techniques comprising many related approaches and methods, and a research field, like how one would consider genomics. eDNA, that is environmental DNA, is the DNA material that is directly obtained from, say, water sample, gut sample, etc., which are sequenced and studied. This approach considers more than just one gene studies like 16S rRNA and provides us with far more detailed functional gene composition. And protocol is relatively simple and economical. It does have its disadvantages though, especially when the sample degrades due to external factors like temperature, enzymes, etc. There are many types of file formats, but we will limit ourselves to the formats that are supported by VSearch. Firstly, FASTA. FASTA is a very simple text-based file format that begins with a greater than character for each read or sequence. This first line is the description. All lines are generally shorter than 80 characters in length. FASTQ is also a text-based format that has, in addition to the biological sequence, its corresponding quality scores. This format usually uses 4 lines per sequence. The first has an @ symbol in the beginning and is followed by a sequence identifier and an optional description, just like a FASTA title line. The second line is the raw sequence. The third is the quality score identifier, which begins with a plus character. It may have the same sequence identifier and any description, just like the first line. The fourth line is the quality values for the sequence in the second line. The number of characters in this line should match the ones in the second line. SAM format is also a text-based format that is sectioned into header and alignment sections. Headings begin with the @ symbol, and this distinguishes headers from the alignment section. Alignment sections have 11 essential fields, which include template name, bitwise flag, template length, etc. K-mers are subsequences of a sequence that are k units long. So if we consider this sequence ATGGCC and we define a k-mer of, say, 3, we get 4 subsequences. Sequence alignment is a huge topic, so we are going to tackle it very superficially. But you can find some really thorough and interesting articles which I have linked in the description box. So, sequence alignment is the arrangement of biological sequences to identify regions of similarity that can help us understand functional and structural relationships between the sequences. Firstly, we have global alignment, which attempts to align every residue in every sequence from start to end. Contrary to this is local alignment, which aligns parts of sequences with the highest similarity. Global alignment can be pairwise with a query dataset and a database set, or can be used as a multiple alignment method, aligning all of the sequences in a given query set. The global alignment generally uses Needleman-Wunsch algorithm, while the latter uses Smith-Waterman algorithm. So these algorithms that I mentioned produce alignments using a technique called dynamic programming, which is basically fractioning a big problem into small subproblems in a recursive fashion. Seed and extend involves seeding, which is identifying exact matches in the read with that of the target. Once this is accomplished, extension takes place from each end to obtain a better match. Heuristic technique is a problem solving method that is used in the sequence alignment. The method juggles speed at the expense of accuracy. Hash table in bioinformatics is a data structure containing multiple lists of genomic positions, one list for each possible KEMA. This is quite helpful in aligning sequences to a genome at a faster pace. Chimera is a contaminant sequence that can be interpreted as a novel sequence, while it is just, in fact, an artifact. This can arise because of sample handling errors or PCR errors, where, due to an aborted cycle, the emerging fragment acts as a primer for the next cycle copying. Oftentimes, chimeric sequences are unfortunately reported as novel sequence. Finally, dereplication is a process of finding unique sequences among a number of copies present. The opposite is re-replication, which is repopulating the dataset with the number of duplicates of a unique sequence as per the original number. Thank you for watching this video. If you found this helpful, please do consider liking, subscribing, and sharing it with your friends and colleagues.